Hi students, coming to the next topic is the strategies for the food development. See, the chapter title strategies means there is in different strategies are there to develop the yield, especially whatever that crops are there, rice production, wheat production, and different types of pulses production, right? So whatever the crops producing products are there, all these products, okay, yield is increased by the following different types of strategies. See, before going to the topic here, we have to discuss about one of the best scientists. He dedicated his life regarding to the plants and crops, the plant, especially crop plants, and their yield. And he is the father of Green Revolution. He is the father of Green Revolution, especially in India. We know Norman Borlaug is the scientist. He is the father of Green Revolution to the world. But best scientist in India, M.S. Swaminathan, he did the enormous research regarding to the plants and the products. Means here whatever that crop plants are there and crop plants producing yield is there, and he did the research, and he born in 1925 August, Kumbakonam. The place is the Kumbakonam. That place is located within the Tamil Nadu. Right? And he is expert. He is one of the best scientists, especially in genetics and the plant breeding. That is nothing but he is the expert in genetics and the plant breeding areas. He knows the remaining also, but he is very best in these two areas. And he done the experiments on Mexican wheat. See, the Mexican wheat research is mostly supported for the Green Revolution. He have that idea on the whole Green Revolution process. Okay, that's the reason why he have the idea. Based on that, he developed different varieties in India. So he did the experiments on the Mexican wheat, and he also supported for a certain part of the Green Revolution throughout the world also. But basically, he is the very popular and very best scientist in India. That's the reason why he is called as father of Green Revolution. And he created one of the high-yielding basmati rice. And father of Green Revolution in India. So these are the points which are related to the Green Revolution process and Green Revolution process done by the M.S. Swaminathan. So all these points are related to, mostly these points, Mexican wheat, this is related to Green Revolution. So this is also related to Green Revolution, this is also related to Green Revolution. Basically, these points are mostly related to the Green Revolution. Green Revolution in India done by the M.S. Swaminathan. That is about that M.S. Swaminathan. And the types of food development. So how the food, different types of foods are produced. Or the types of food development process are there. We know that RNA technology, recombinant DNA technology. See, certain desired genes are isolated, which are cutted and inserted into the TI plasmid. That TI plasmid inserted into the one of the plant. Again, from that plant cells, we are creating the new plant, or we are producing the new plant, and the plant has the desired characters. The desired characters are nothing but high yielding capacity, high disease resistant, and the nutritional value is more within the yields or within the products. Right? So these are the different points, and some of the plants also having that disease resistant, maybe the pathogenic resistance, maybe some of the abiotic stress conditions, maybe some plants grown at high temperature, even absence of water also, some are grown in salinity conditions also. See, that kind of plants we are creating by the usage of RNA technology process. And next is the plant breeding. So what is plant breeding? Simple. In this case, we are selecting the two parents. The parents are having desirable characters. One is high yielding producing plant, other one is the high disease resistant representative. So two different plants, and we are doing the hybridization between these two plants. We are doing hybridization between these two plants. Right? Once if we did the hybridization between these two plants, maybe in the next generation, the plants are produced. The plants are having both the characters. The plants are having both the characters. So that is nothing but plant breeding technique. Right? By the usage of plant breeding technique also, we are producing different types of plants. And as well as the mutational plant breeding. Mutational plant breeding is nothing but some of the plant seeds are there, or the leaves are there, or the plant pods are there. The plant pods are exposed to mutagens. Mutagens are nothing but mutation causing agents. Mutation causing agents which are called as mutagens. So different types of mutagens are there. Maybe the seeds are exposed to the UV rays. Maybe the leaves are exposed to the UV rays. The mutations are generated. The mutations are generated. Then, see, some of the mutations are not useful. Some of the mutations are harmful. Some of the mutations are useful. So, whatever that mutated plants are there, within the mutated plants, whatever that high yielding producing plants are there, means the plants are having useful mutations. The plants are having useful mutations. That kind of plants are selected and we are using. We are cultivating, right? That is about that types of the food developments. One is the RDA technology, plant breeding, and the mutational plant breeding, right? And crop yield. See, we know there is two types of the crop cultivations are there. One is that traditional crop plantation. Traditional crop plantation, normal crop plantation. Whatever the seeds we have, the seeds we are simply using to cultivate. Any seeds, what seeds are there? Okay, we are not selection, we are not following the selection, we are not doing any particular characters. Whatever the seeds are there, we are simply shown to the field, we are cultivating the plants. So, traditional crop plantation, that is nothing but traditional crop plantation. So, within that traditional crop plantation, within the traditional crop plantation process, increase the cultivation area. Increase the cultivated land. Increase the cultivated land. So that is about the traditional crop plantation. But coming to the next one, green revolution process. See, here there is a particular characters are desirable. The seeds should have, the plants should have the desirable characters, high yielding capacity, high disease resistance, and even these can survive in the drought conditions also, right? And triple the yield, especially within the green revolution process or by the green revolution process, triple the yield, not double, triple the yield, right? That is about the green revolution process, right? And coming to the next one, plant breeding. There is a conventional plant breeding is there, classical plant breeding is there. So conventional plant breeding started with the human civilization process. In and around, this is the oldest method, and in and around 9,000 to 11,000 years back, 9,000 to 11,000 years back, okay, the plant cultivation is started. The plants cultivation is started. So started with the human civilization. Started with the human civilization. So 9,000 to 11,000 years back, okay, the plant cultivation is started. And not for the desirable character. Simple. Here also there is no desirable character. Simply plants are cultivated. Simply plants are cultivated and harvesting the yield. Harvesting the yield. That's it only. That time farmers not have much knowledge. When the days are going on, that time farmers have the idea of the crop cultivation. So somewhat the cultivation process is developed. The cultivation process is developed. But initially, okay, the plants, whatever that the crops are there, the crops cultivated simply, not for the desirable characters, not for the desirable characters. And as is the classical plant breeding process. Simple. This is the advanced process. This is the advanced process. Here, priority is given to the desirable characters. Priority is given to the desirable characters. So, here pure lines are selected. Pure lines means always these are maintained the homozygosity. Always these are maintained the homozygosity. And remember, these are having desirable characters. The plants having desirable characters, with homozygosity, right? And genetics are there, molecular biology is there, tissue culture is there, right? Genetics, molecular biology, tissue culture. These three are the best techniques. So this is advanced method because in this case we are using advanced techniques. Genetics we are following genetics, and we are following molecular biology. We are using the tissue culture. So presence of these advanced techniques, we are producing the high yield producing plants, high disease resistant plants, and the plants having the abiotic resistance, means the plants have resistance to cold, the plants have resistance to high temperature, the plants grown in different types of soils, the plant grown in high salinity regions, right? So all these are shown by the plants which are produced by the classical plant breeding, right? And increase the yield, produce the disease resistance and abiotic resistant plants. We know yield is increased, obvious thing, and next one is the produce disease resistant plants and abiotic resistant plants. Disease resistant plants and abiotic resistant plants, right? That is about the types of the plant breeding process. One is the conventional plant breeding, other one is the classical plant breeding. And all of you remember the next topic, we will discuss about the steps in plant breeding process, or the process of plant breeding. We will discuss now. And coming to the next topic is the steps in plant breeding process. See, basically here 5 steps are present. And we have to discuss about these steps. And remember, those who are want to go to plant research, this process is very, very important, and this is a very basic process to produce the hybrid plants or the new variety of the plants. See, the very first step is the collection of variability. Generally we know crop plants. Whatever the plants we are cultivating, these are called as crop plants. In crop plants, different types of varieties are there. In crop plants also, some are having high yielding capacity, some are having high disease resistant, and some are having low disease resistant, and some are having good nutritional value. So these are the different types of crop plants. And if we think about the wild plants, some are grown naturally, wild plants also there. So see, we are collecting the combinations. Or we are collecting the different types of desirable characters. See, pre-existing genetic variability available in the wild crops. Whatever that wild crops are there, within that wild crops, pre-existing genetic variability. Whatever that expecting characters are there, the characters are available within the wild crops. And prerequisite natural genes. Within the crop plants, some of the characters are naturally present. Right? So here genes collection from the crop plants. Simple. Whatever that desirable characters which are present in crop plants, whatever that desirable characters which are present in wild plant, we are collecting. So the collection process, the collection is nothing but maybe we collect the pollen grains, maybe we collect the leaf of the plant, maybe we collect the different parts of the plants. If we have the plant parts, we can produce a new plant because we have the tissue culture. By the usage of tissue culture, we can produce the new plants. So here, whatever the parts we are collecting from the wild plants, desirable characters contain wild plants, and the desirable characters contain crop plants, which are stored. See, the process is called as germplasm collection. So what is germplasm collection? Simple. Desirable character containing plant pods are collected, which are subjecting for the cryopreservation process. When we need that time, the plant pods are activated and utilized. See, we are subjecting for the cryopreservation process. So this step is called as germplasm collection and germplasm storage. Storage done by the cryopreservation. cryopreservation process. And next is the evaluation and selection of parent plants. So different types of parent plants are there. Different types of parent plants are there. Within that parent plants, what are the characters required to the farmer, or what are the characters are expecting the scientist in the next generation? So that characters contain plants are selected. That characters contain parent plants are selected. So here germplasm evaluated, identified desirable combinations. See here what I have desirable characters are present. For suppose in one plant high yielding capacity is there, in another plant high disease resistance is there, right? So this is the one combination. See high yielding and high desirable, high yielding and high disease resistant. Both the characters are expressed in one plant. That is very useful. Very useful. So some of the plants, disease abiotic resistance is there, that can the plant can tolerate the high temperature, right? And the plant low yielding capacity. Then the plant is hybridized with the high yielding plant, high yield producing plant. So we are doing hybridization between these two plants, right? And selection of parental plants. In all these, whatever that desirable content plants are there, we are selecting. Produce the pure lines. Sir, what are pure lines? What are pure lines? See, pure lines are nothing but homozygosity content plants. Homozygosity content plants. See, we know this is capital T, the both are same. This is homozygosity. And capital T, small t, this is heterozygosity. And small t, this is homozygosity. So, once the parental plants are selected and which are subjecting for the production of pure lines, sir, why we are producing the pure lines? We are maintaining the homozygosity. What is the use? Okay, if we maintain the homozygosity. See, if we maintain the homozygosity, whatever that desirable characters are there, the desirable characters stable. The desirable characters are stable in the next generation also. The desirable characters are stable. That's the reason why we are producing the pure lines by the self-pollination process. By the self-pollination process, we are producing the pure lines. Right? That is about that evaluation and selection of parental plants, and cross hybridization among the selected parents. Whatever plants we are selected, plant 1 and plant 2, two plants are selected. See, according to the NCRT, one is the high protein yielding capacity, other one is the high disease resistant. Two different plants. We are doing hybridization between these two plants. So how we are doing hybridization? See, one of the plant is considered as male plant and other plant is considered as female plant. So from the male plant, pollen grains are selected, and see, we are going for the artificial hybridization process. We are adding the pollen grains to the stigma of the female plant. Then between these two, pollination will be takes place. Then the seeds will be produced. Fertilization takes place, seeds will be produced. See, this is one of the artificial pollination process. See, we know in this case, in the first year we have studied about the emasculation process, bagging and rebagging. All the techniques we can apply here. There is an emasculation, scissoring the anthers, bagging process, and next one is the addition of pollen grains, right? And next one, again, the bagging will be takes place. Right? All these techniques are applied to the hybridization process, and this is a time-consuming process and tedious process. This is the time-consuming process and tedious process. See, for suppose for the production of hybridized plant or desirable character content plant. See, generally, sir, within how many number of plants? Okay, we can identify one hybrid plant. For suppose I produced here few hundred plants to few thousands. Okay, few hundred hundreds to few thousand plants. See, I produced, okay, within laboratory, within that research area, I produced few hundred plants or few thousand plants. For suppose I produced here 1 lakh plants. Maybe within 1 lakh plants, one plant is the hybridization plant. Maybe one plant is the hybridization plant. Maybe 2 plants are the hybridization plant. That's good. Okay. See, okay, hybridization plants, all plants are not hybridization plants. Only very few plants are hybridization plants. So this is the time-consuming process because we need to cultivate all the plants and we need to evaluate all the parental plants. And the tedious process we need a particular order, right? So that is important because we did the hybridization, we don't know in the next generation both the characters are expressed or not. We need to wait and we need to evaluate. And the next generation also we need to do the research. The next generation also we need to do the research because the characters are stable or not in the next generations. We have to do, right? That is about that hybridization process. And then next one is the selection and testing of superior recombinants. Selection and testing of superior recombinants. Simple. A selection of hybrids with desirable characters. Whatever that hybrid plants we are producing, okay, we are produced here, we are selecting. After the hybridization process, we are selecting here. And scientific evaluation, I already said scientific evaluation is required. Simple. We planted one hybrid plant, okay, day 1, what are the characters, day 2, day 3, day 4, okay, after 1 week, after 2 weeks, when the flowers are produced, colors of flowers, okay, and yield, how much yield is produced. Then, okay, plant 2, what are the characters are there? Plant 3, what are the characters are there? Plant 4. So, we need to evaluate all the characters of these hybrid plants, right? And the scientific evaluation is required. And self-pollination, again, once we selected one of the hybrid plant, okay, within that plant, again we have to follow that pure lines production, pure lines produced by the self-pollination process, right? Because we are stabilizing the desirable characters. We are stabilizing the desirable characters to the next generations also, right? And the last one, testing, release and commercialization of new cultivars. Simple. What are the plants we produce? The plants are having all the desirable characters or not? We check it here. The next one is, these plants are grown in all the types of soils or not? For suppose, within Telangana region, the grape plants are grown very rapidly and high yield is produced. Within our area, okay, the grape plants are not properly grown, which gives the low yield. Right? And one of the best example, within our area, rice plants are very rapidly grown. Within the Telangana region, the rice plants are not rapidly grown. So, soil to soil, the growth of the plants will be different. So, here disease resistance also different. Soil to soil and climate to climate, the disease resistance also different. Okay, we need to check all these parameters and perform under ideal fertilization. For suppose if we use one type of fertilizers on a particular ratio, then maybe that will give the high yield. If I use another type of ratio, maybe gives the low yield, right? Fertilizers and the irrigation process and the crop management, we need to check all these parameters, okay, towards the particular hybrid plant cultivation, right? And the this is one of the plants, plant 1, plant 2, plant 3, plant 4, and the survival. See, there is in different types of soils. There is plant 1 soil, plant 2, plant 3, plant 4, different soils which are grown in different soils. Survival, maybe plant 1 is grown, maybe plant 2 is not grown, maybe plant 3 is grown, maybe plant 4 is not grown. Okay, we don't know, just can explain because some plants grown in some soils will growth. Here, growth is rapid. The first case growth is rapid. Here, growth is poor. No growth here because the plant is like growth is there. The plant is like okay, in certain cases slow. And here, moderate. Here, growth is moderate. Right? Here, growth is maybe no growth because maybe the seeds are not germinated. Right? Yield. Here high yield is there, here high yield is there, here low yield. This is here plant is right, here low yield, right? Here yield is slow or low yield, sorry, not low yield, right? Here also no yield. Here also no yield, right? So these are the different types of parameters we are checking. See, these parameters have changed from one climatic condition to another climatic condition, from one type of soil to another type of soil. So by this way we are producing the different types of the hybrid plants by the plant breeding technique. Next one.